Hi, I'm Jacob Lamberth. I'm an R&D scientist in the genome and epigenome editing group of MilliporeSigma. And I'm happy to talk to you today in this LabRoots oncology webinar about some of the work we've been doing here and to show you some exciting new technologies and techniques. Specifically, I'm going to tell you all about highly efficient and specific genome editing with Cas9 Nickase Ribonucleoproteins in primary T cells. We've recently submitted a paper of this data to the bioRxiv, but I'm going to take you through it all today. This is a combination of a multitude of different technologies and advances, so let's go over the components. First, let's talk about what are Cas9 Nickase Ribonucleoproteins. Starting off with the most basic of basics, I know that most of you will be familiar with all of this already, but just in case, I'm going to give everyone a quick rundown of CRISPR-Cas9, what it is and how it works. The most common description you see in all of the media headlines is that they are molecular scissors used in gene editing, which is pretty accurate. CRISPR stands for clustered regularly interspaced short palindromic repeats. But what does that mean? It's a native system found in many bacterial species that functions as an adaptive immune system against invading viral DNA. In the native bacteria, CRISPR systems identify past invading foreign viral DNA and cleave it, rendering it harmless. But this cleavage has been exploited in eukaryotic cells to create RNA-guided site-specific genetic manipulations. But what exactly does the cleavage? Those are Cas or CRISPR-associated nucleases. As you can see here in this diagram, the nucleases are guided to a specific DNA sequence by a complementary guide RNA. Once at this target, the two cleaving domains, showcased here in the diagram, RuC and HNH, cleave each strand of DNA at the target site, forming a double-stranded break. Any resulting genetic change relies on the host cell's repair machinery. In mammalian cells, either non-homologous injoining or homology-directed repair. The first causes random short insertions and deletions, and the second utilizes a repair template that the CRISPR users supply to fix the break more precisely, resulting in integration of a sequence. Cas9, this specifically, is currently the most popular variety of the Cas nucleases, SpCas9, from Streptococcus pyogenes. But it can have off-target cleavage activity with enough complementarity between the intended site and a genomic site at another location. In order to deal with this off-targeting issue, there are several different routes that researchers have taken to reduce these effects. One such route is using a paired Cas9 Nickase system. Cas9 Nickase is a mutated version of Cas9 with a D10A mutation, which renders the RuC cutting domain of SpCas9 inactive. By doing so, the full targeting and efficiency of the nuclease is retained, while only a single-stranded cut in the DNA is made. The nuclease can still find and cut the target site, but only one strand of DNA is cut, and we call this a nick. With this change, however, different cellular machinery is activated to repair the single-stranded break, base excision repair. Instead of NHEJ or HDR, which causes insertions and deletions, the single-stranded break repair perfectly repairs the DNA as the correct template is always there on the other strand. In order to combat this, you have to use a nickase to somehow induce double-stranded break repair, which is where the paired nickase strategy comes in. To do this, you need to design two guide RNAs with a 5' to 5' orientation, as shown here, and have the PAMs ranging from 30 to 150 base pairs apart. By doing so, you will have one nickase cut the top strand and the other cut the bottom strand. Doing this within this base pair window has been shown to trigger double-stranded break repair mechanisms instead of just single-strand repair. This method combats the off-targeting of SpCas9 by requiring pairs to come together to nick together in order for any editing to occur. Any off-target nicks in the genome will not have the adjacent other nick, and only the single-stranded mechanisms will trigger, where no editing can occur. The probability of off-targets meeting in this paired criteria is extremely low in the human genome. Therefore, this is a highly specific method of genome editing, and it has been around for a couple of years. Another newer method that we combine this with is to use Cas9 Ribonucleoproteins. Traditionally, Cas9 nucleases are delivered via plasmids or mRNAs that encode for the protein and guide sequences. These methods rely on host cell machinery to transcribe and/or translate. They have been tailored to function in each specific cell type. A ribonucleoprotein is a recombinant Cas9 protein that has been complexed with the targeting guide RNA before delivery to cells. There are traditionally many problems that occur when you use plasmids or mRNAs. There can be random integration of DNA into the host, which is unwanted if you want to do more than just focus on the guide site. There can also be toxicity issues. Many cell types reject and undergo apoptosis when introduced to foreign DNA, especially primary cells. And this toxicity can also provoke immune responses in the host organisms. Also, when you introduce plasmids or mRNA, there will also be continual expression, which increases off-target probability. The cellular machinery will continue to express the Cas9 and guide that you introduce to the cell. Well, this is great for making sure that there are plenty of copies for cutting your target site. The extreme excess of copies dramatically increases the possibilities of a partial match occurring somewhere along the genome, increasing the likelihood of an off-target genetic effect. You can combat this by using an RNP. They are much more universal. They will work with any cell with DNA. There's no need for expression, as the only thing you'll have to design is the target itself. They also cut immediately after delivery, as they bypass transcription and translation. They are ready to find the target site and cut immediately after delivery. They also rapidly degrade. The continual expression issues of the other methods aren't something one has to worry about when working with RNPs. They don't stick around long enough to find much more than the exact sequence before they are degraded by the cell. These are rapidly becoming the new standard method of delivery, especially in primary cell types. So we decided to make a Nickase version of an SP Cas9 RNP. But the first thing we had to do was make sure that it worked properly. Before we did any advanced testing in cells, we first had to verify their cleaving abilities in general. We needed to make sure that the D10A mutation was properly made and that no double-stranded cleavage could occur. To do this, we did a simple in vitro assay. We incubated the Cas9 Nickase RNP complex with a supercoiled plasmid with the designed target. A double-stranded break would result in a linearized plasmid, and a nick in the plasmid would cause it to unwind and run slower on a gel, and can be seen higher than the supercoiled plasmid, which is exactly what can be seen here in our tests with an average efficiency of 90%. So we now know that we were able to successfully combine the Nickase technology with the RNP technology. Next, we needed to determine if it can work in human cells. For this next proof of concept, we worked on human K562 cells in a mortal leukemia cell line at the EMX1 locus, which is a transcription factor and a common CRISPR positive control site. We chose this cell line and locus because in our hands, the cells are extremely easy to culture and transfect, and the locus is open and available to edit in. Next, I'm going to go over how we actually performed the experiments and analyzed the data. While there are a multitude of different methods to get RNPs into cells, we worked with our collaborators' MaxCyte flow electroporation system. These base steps are outlined here. You briefly incubate the Cas9 and guide RNA to form an RNP. You mix the cells suspended in a transfection solution. You then use the machine to zap the cells, punching holes in the cell membranes and allowing RNPs to enter. Once you allow the cells to recover, you can then harvest the genomic DNA. Another method we used was Lanza's nucleafection, which is a transfection method that has a similar workflow. Once we have this genomic DNA, we then needed to determine whether any editing took place. Historically, we have used the cell-1 surveyor assay to determine CRISPR editing in cell lines. A brief run-through of this is that once you have the genomic DNA, you amplify a region surrounding the target site of interest via PCR. You then melt and re-anneal the PCR product, and by doing so, heteroduplexes will form, as diagrammed here. These create bubbles in the DNA strands where mutations cause mismatches. You can then digest the product with the surveyor nuclease, which cuts single-stranded bubbles, cleaving the DNA strand at the mismatched sites, allowing us to calculate mutations as indels, or insertions and deletions, after visually analyzing a gel, as example here. We followed these exact steps targeting the EMX1 locus in K562 cells. And here are the results of our first testing. We tested 6 different conditions. First, our cells alone. Second, our cells nucleaffected with wild type Cas9 RNP with cRNA. Then wild type again with cRNA-B. Next, our Nickase RNPs were nucleaffected together with cRNA-A and B. And the next 2 are our Nickase RNPs nucleaffected alone. Key takeaways from these results are that 2 single guide wild type Cas9 nucleases have indel percentages of 23 and 10%, and that the paired Nickase RNPs were able to cut at a similar 22%, meaning that the Nickase RNPs are on par with the current existing RNP technology. Another important takeaway confirms the in vitro experiment from earlier, in which the unpaired Nickase RNPs have 0% cleavage efficiency, meaning our Nickase RNPs are only creating single-stranded nicks and are unable to produce indels alone. This is the ideal proof of concept, but it was in an ideal situation, with strong editing seen on both wild-type RNPs at known strong controls. Next, we wanted to see whether or not the system would work if not every guide was a strong editing guide. This is the same general setup as the previous experiment, with one of the wild-type Cas9 RNPs cutting at about 20%, but the difference in this one is that the other guide in the pair has little to no visible editing with wild-type Cas9. These results, while still showing no editing the paired nickases, however, do not show us clear editing with the paired nickase. There is a smear, as indicated by the red bar on the gel, but I believe due to the limitations of the cell-1 assay, gels cannot accurately show what is happening on the DNA level. To remedy this, we decided to do more sequencing-based analysis after this to determine whether editing actually took place. There are 2 main types of sequencing-based analysis. One involves comparing Sanger sequencing reads, in which PCR products are sequenced as a pool, and the computer reads overall fluorescence of the pool as a whole to determine each nucleotide. The other is next-generation sequencing, in which each PCR product is sequenced individually and the exact sequence of each strand, mutant and wild type, is known. The basic steps of NGS are to PCR amplify the region surrounding the target site of the genomic DNA, just like the other methods. You then, however, deviate and prep the PCR product with specific adapters for NGS sequencing before running them on a specialized sequencer. Specialty bioinformatics tools are then used to analyze results and determine the nature and quantities of each mutation type. This is a much more accurate method of analysis than cell-1, so we decided to use the same genomic DNA of a few of the samples of the previously unclear cell-1 results and then performed NGS on them. This is a mutation frequency plot of the previous cell-1 data, which shows the percentage of editing at each base pair along the sequencing read. Controlled DNA reads are in green, wild-type SpCas9 edited DNA reads are in blue, paired Nickase Cas9 edited DNA reads are in pink, and unpaired Nickase Cas9 edited DNA is in yellow. This NGS analysis showed us that yes, this Nickase guide pair was functioning correctly. It was just that our assay was unable to recognize it across both guide sites. There is on average 10% editing with the paired Nickase nucleofection. Another big takeaway is, as you can see, the wild-type SpCas9 has very strong editing at a single spot, which shows up clearly on the cell 1 assay. But the Nickase editing is much more spread out, showing up as the smear on a gel. We believe this can allow for editing events along a much wider window than just a single guide alone, perhaps encouraging larger knockouts. Incidentally, it also shows that the unpaired Nickase leaves the DNA completely unedited, with every base pair along the cut site being completely identical to the unedited wild-type DNA, proving the specificity of our system at the nucleotide level. Because of the dramatic increase in data fidelity, from here on out, we used sequencing-based analysis on all important results. We repeated this experiment in triplicate, and we're excited to see editing in NGS that was not seen in the cell 1 assay gels. Not only that, but our paired Nickase RNP design was able to edit in a region that wild-type SpCas9 was not able to edit in well, as 10% is our usual cutoff for an effective cutting guide. And in addition, when alone, leaves the DNA completely unedited. This test was another great proof of concept for the technology. We were able to show paired Nickase system working well at multiple different locations, as well as confirming that any possibility of off-targeting is negated by showing that even at the strong on-target location, where wild-type SpCas9 cuts at 40%, there is no editing seen when the unpaired Nickase nicks at that location. If a perfect guide match at a strong location results in zero editing with an unpaired Nickase, any imperfect complementarity to the guide at an off-target site will result in zero editing as well. The combination of efficiency and specificity make this technology incredibly useful for therapeutic applications. So we wanted to test to see how well they can perform at a more relevant target on a more relevant oncology cell line. For this, we decided to test the system in CD8 positive primary T cells at the PD-1 locus. As I'm sure you're all aware, PD-1 is a clinically relevant target in oncology. It has been shown that a way cancer cells evade detection is to utilize immune checkpoint molecules to circumvent an immune system response. T cell would see these and assume that the cancer cell is normal and go dormant. One such negative regulator of T cell activity is the ligand PD-L1, found on tumor cells, and it functions by the binding of programmed death 1, or PD-1, a receptor on activated T cells. The anti-tumor effect of T cells can be improved by blocking the PD-1 receptor, which has been shown to increase the immune response against cancer cells, and it is a safer and more effective technique than attempting to act on the cancer itself. This is known as checkpoint inhibition, and in doing so, increases the wakefulness of the T cells in regard to cancer cell fighting. This checkpoint inhibition is a very well-known technique, and it has been done with antibodies, ZFNs, TALENs, and even other CRISPR systems. However, each method has had known drawbacks, be it price, ease, or specificity. Most, if not all, of these drawbacks can be solved with our new paired Nickase RNP system. So we went to work and designed some guides that target PD-1. We wanted to quickly test to see if the system was functional and that the guides we designed for PD-1 were able to work. So we did a quick experiment in K562 cells. This is a cell 1 gel that shows that each guide achieves 1 to 15% indels alone with wild-type SP-Cas9. But interestingly, we see that they appear to function even better as a paired system, with 17 to 25% indels in the paired Nickase system, even when using the 1% editing guide. This is a very promising initial testing of the target, but we needed to move forward onto the testing in CD8 positive primary T cells to see if the editing can be functionally used. Up until this point, we had been using a 2-part synthetic guide RNA in our RNP systems. There are 2 different forms of synthetic guide RNA you can use, a 2-part or a single guide. In the 2-part system, a universal tracer RNA and a target-specific cRNA are annealed before being complexed into a single guide, while in the single guide system, the entire guide RNA is synthesized in one piece. Both can be modified to increase efficiency and stability. Historically, the 2-part system is cheaper to synthesize and works well in immortal cell lines in our hands. But as we tried to move these experiments into primary cells, we were unable to get any of the indels we had previously seen. The 10 to 20% indels that we had previously seen in K562 cells dropped to near undetectable levels. Recent papers have shown that chemically modified single guide RNA RNPs have enhanced Cas9 editing in human primary cells. So we've repeated these experiments with the new modified single guides to verify this claim. The difference in the results with the new guides in our primary T cells was staggering. From seeing little to no editing whatsoever in any of our previously very strong guide pairs, to similar and even better indel percentage than what was seen in immortal cell lines, up to 50% from sub 2%. With this new addition, our paired Nickase RNP system was fully functional in primary cell lines, so we could go forward with a full panel to see their editing potential. Here is the same data made with single guide RNA modified, but with the full panel, including wild-type SP-Cas9 in triplicate. We see strong cutting above 10% across the board for all conditions, with one paired Nickase having the highest editing of the lot. This is a much more relevant proof of concept than EMX1 in K562 cells. Our paired Nickase system is now working just as effectively as the wild-type SP-Cas9 system in PBMCs. The next test would be confirming that the specificity of the system that we saw in K562 cells carry over, which is exactly what we did. Our team tested the SP-Cas9 Nickase with only the single guides and then the 2 paired together. And the results here confirm that 0 editing is taking place when the Nickases are transfected alone in a primary cell line, proving that the specificity we saw in immortal cell lines translates to primary cell lines as well. Putting all of this together, we now know that we have a highly specific editing system with high cutting efficiency in a relevant cell line at a relevant clinical target. The next step is to determine if the editing being performed by this system is actually phenotypically knocking out the protein of interest and not just insignificantly genotypically editing the sequence. We worked with our collaborator, MaxCyte, to perform PD-1 immunostaining and flow cytometry analysis to determine the expression levels of PD-1 after being edited with SP-Cas9 with our paired Nickase system. In triplicate, they transfected RNPs into cells with their flow electroporation system, let the cells recover, and performed analysis at 3 time points. What you can see here is that at all time intervals, there is a greater phenotypic knockout of PD-1 in PMBCs using the Nickase system than using SP-Cas9 alone, even though the genotypic indel percentage was comparable between the 2 conditions. You can see that the knockouts after 5 days in the wild-type SP-Cas9 system have up to a 40% drop in expression, while in the paired Nickase system, that's more than doubled, with over an 80% drop in expression compared to control levels. We hypothesize that this is because, as we saw earlier in the mutation frequency plot, paired nickase editing spreads out its indels across a much wider area than a single guide system, leading to larger knockouts, causing more of a phenotypic disruption than wild-type SB-Cas9 does. This makes it an even more effective tool for therapeutic purposes than we had previously hypothesized. However, when talking about phenotypes, we next had to make sure that the Nickase system cannot cause off-target phenotype effects in a CRISPR-I fashion, even if the Nickase itself has repeatedly shown that it does not edit DNA at the genotype level. We tested this in the same fashion as the previous test. MaxCyte transfected the Nickase RNP with a single guide alone and waited 120 hours before measuring the PD-1 expression levels in the cells. What you can see here is that in an unpaired Nickase system, after 5 days, the expression levels are identical to the wild-type controls. This is at the same time point and conditions that the paired Nickase system dropped expression levels by over 80%. Repeating this in triplicate verified that the system remains highly specific even on the protein expression level. Putting everything together again, we now have a CRISPR-Cas editing system with high efficiency in both indel percentage and phenotypic knockout, without the possibility of any off-targeting effects on the sequencing or gene expression level. Exactly the type of system a scientist in the therapeutic realm needs to ensure the only thing they're changing in cells are what they designed it to do. We wanted to expand our scope and test this new system in a variety of immunotherapy targets in T cells to ensure that the system is just as flexible as wild-type SB-Cas9 is. In order to do this, we looked through different checkpoint immunotherapy literatures and found 3 other candidates for protein targets that have potential therapeutic significance, and designed our guides to target these loci: CTLA4, TIM3, and TRAC. We went through all the preliminary steps as before for validation, testing in immortal cell lines and such before moving forward into testing in CD8-positive primary T cells. What you can see here are the results of these tests performed in triplicate. While the editing efficiency varies from target to target, as all Cas9 systems do, our paired Nickase system gets significant editing results across the board, and the high specificity of these results will carry over, making it an excellent choice for future CRISPR immunotherapy applications and research for scientists who want to have high efficiency and specificity in their results. Thank you for listening. You can read more about our work and see these results more in depth in our paper that was recently submitted to the bioRxiv. Thank you.